So, good afternoon, everybody. So, we are very delighted to have Hirak with us for this departmental seminar. And let me very briefly introduce Hirak, who is a bit junior to me and also a friend to me. Hirak did his master's from Jadavpur in 2000, and then he completed his post-MSc associates and then consequently PhD, mostly working on biophysical chemistry from SIT Kolkata. And then he moved to University of North Carolina, Chapel Hill to do a postdoctoral research. And when he returned to India in 2012, he joined the CCNB as a CSIR pool scientist. And in 2004, he eventually joins the Sambalpur University as a UGC-Assistant professor, and he is still there. He has published more than 58 papers and 4 book chapters, mostly working, recently he is working on membrane biology, as I understand. And he received several awards, and most recently he has been awarded with the ACRB Science and Technology Award for Research. So today, as I understand, Hirak is going to talk on some lipid composition in membrane fusion, and which plays a key role actually manipulating the lipid-peptide interaction. So Hirak, you just start your talk, and maybe do you mind getting questions within your talk, or we should put it in the end?
No problem.
Okay, so if anyone has any question, he or she may raise the hand or put it in the chat box, and we will communicate with Hirak. Okay, Hirak, fine?
Okay.
Okay, so please, Hirak.
Oh, so thank you, Rajivda and Suman, for really having me in this platform for departmental seminar. And just let me know if my signal drops or some problem. Suman has my number, can just drop a WhatsApp message.
Sure, I'll do that. No problem.
Yeah. So let me share my screen. So I think my screen is visible.
Yeah, it is coming.
No, somehow it's not visible, no? Suman, I cannot see
it. Not yet. It is not. Maybe you can close the presentation and share again. Yeah. Now it is here. Now you can see. There is a lag. Little bit of lag is there, I think.
Yes.
Yeah. So I think it's visible now, no?
Yes, it is visible. Please continue.
Oh, just let me do one thing. I will close my camera.
Yes, that will be better. You close your camera.
Yes. Okay, so thank you once again for joining. So today I will be talking about lipid composition in membrane fusion, a key player in modulating lipid-peptide interaction and beyond. So what we mainly know that lipid basically modulate the lipid-peptide interaction, and that has an impact in fusion in other activities of any membrane protein or peptides. But I will just put some emphasis on this beyond part. So it's not just lipid-peptide interaction, it does many more than what we expect. So in short, the membrane fusion is a process by which two initially distinct lipid bilayers, they merge their cores, resulting in one interconnected structure. Like this blue vesicle, so this is a membrane containing blue and yellow, so they go through some intermediate structure, and finally this blue has to mix with yellow to give a green color. So unless and until this vesicular or intermembrane liquid they mix, we don't call it full fusion. So it's like if you take 2 soap bubbles and if you bring them very close to each other slowly, then it will form a big bubble. So if you bring it very quickly, then it might rupture. So this process is highly fine-tuned process so that these two membranes, they do not burst, rather they fuse and forms one interconnected structure. And in biology, there are several proteins which basically helps to occur this fusion process in a very sophisticated, specific, and subtle way, so that these membranes, they don't burst, rather they fuse with one another, and the inner material from one membrane can enter into the inner material of other membrane. So but why do we need this process? So if we see, there are many constitutive biological processes like trafficking, compartmentalization, import and export of nutrients and waste, vesiculation, intercellular communication, cell division. They all depend on this process. But in our lab, we are mainly focused in the entry of lipid enveloped viruses. So from this corona pandemic, everyone is having a good knowledge on the entry of virus and the shape and structure, and we all know that it's basically the RNA material is encapsulated by lipids, and that's why that who suggested to wash our hands with soap, so that the lipids will be dissolved and virus will not be able to enter into the host cell. So we are mainly interested in the entry of this lipid encapsulated virus. So this lipid encapsulated virus, they basically fuse with the cell membrane or with the internal organelles. Like influenza, it fuses with the endosome, not at the cell surface. But there is a fusion between the viral membrane as well as the host membrane, and resulting the viral inner material that gets transferred to the host cell. And then this DNA or RNA, depending on the type of virus, they use the host cell machinery to replicate their genetic material. So what we know about this process, that I showed you those threads in my second slide, that proteins, they basically help to membrane to come close to each other and fuse to membrane. So basically these proteins are known that they form a trimeric structure. So this is a very classic model of influenza hemagglutinin, and the structure has been solved by crystallography. And this crystallographic structure only lacks the fusion peptide domain, but it has the other part. And it showed that the fusion protein is basically a trimeric structure. And then what happened? This transmembrane domain, which basically resides on the viral membrane, and the other part is the N-terminal part, which we call fusion peptide, that basically enters into the host cell. So for influenza, it's endosomal membrane. So this N-terminal fusion peptide, it basically creates the contact between the viral membrane and the host cell membrane. And then this triple helix, this is a triple helix with the N-heptide region of the protein, and there is a triple helix with the C-heptide region of the protein, and then they interact with each other and forms a 6-helix bundle, and that basically helps to bring this host cell in contact with the virus membrane, and subsequently it fuses with the host cell. The viral membrane fuses with the host cell. So this is very crucial. This structural conformational change is very crucial, and it is believed that this conformational energy actually provides the energy to cross the activation barrier for the fusion. So, and the same thing we also observed, this trimeric structure also we observed for other proteins like this SARS-CoV spike protein. So this SARS coronavirus 2 spike protein, this is a cryo-TM structure, and there they also showed it forms a trimeric structure. So if you see even the GP41 of human immunodeficiency virus, HIV, that protein, the protein, it's not the entire protein, but the protein without the fusion peptide and transmembrane domain, thus we have the crystallographic structure for that protein, and that protein is also trimer. So we recently wrote a book chapter that this oligomerization of fusion proteins, a common symptom for class 1 viruses. So this trimerization is very crucial. So I will come to the importance of the trimeric structure in my later slide. So just I want to tell you this oligomerization is very crucial. So if we see the stock model, lipid stock model, it says that the fusion protein, it has a receptor binding domain and a fusion domain, and the receptor binding domain, these lines, so they basically interact with the host cell receptor, and then we see there is a cleavage between the receptor binding domain and fusion domain, so that the fusion domain, it opens up, and the N-terminal peptide, which we call fusion peptide, it basically enters to the host cell, and then conformational change takes place, and that conformational change basically creates a bend at the membrane, in both membrane, and which subsequently come close to each other and forms a stalk-like structure. And this stalk, it undergoes via different conformational intermediates like transmembrane contact, extended transmembrane contact, and ultimately they get separated and we get a cleared fusion pore. So in more detail, the cartoon structure, that all fusion protein, it has a signal peptide at the very end terminal and then receptor binding domain, and this is basically the cleavage site between the receptor binding domain and the fusion domain. And if I look at the fusion domain more detail, so it has a fusion peptide at the end terminal of the fusion domain, followed by the heptide region, transmembrane domain, and cytoplasmic tail. But we will focus mainly on the fusion peptide, which is basically 20 to 25 amino acid long N-terminus fusion domain that plays a crucial role in fusion. And the interesting thing is that mutation in the fusion peptide demonstrates same phenotype as that in fusion protein. So that basically establishes the importance of fusion peptide and studying the process just with a minimalistic model where we have only the fusion peptide, because the mutational study, they match with the total protein mutational study. And interestingly, fusion peptide itself is capable of inducing fusion between lipid vesicles, and several putative mechanisms have been proposed on how these fusion peptides actually promotes fusion. So when we started our lab, we looked up literature, and there is a beautiful paper by Lucas Tam group from University of Virginia. What they showed, they basically studied with the GP41 from HIV virus, and this GP41 fusion peptide, it is pure alpha helical, the red one, when membrane does not have any cholesterol. But if you increase cholesterol concentration in the membrane, the peptide, it moves from alpha helical to beta sheet structure. And then what they did, they basically studied lipid mixing as well as content mixing, and which basically represents the fusion. And they observed that as they increased the concentration of cholesterol, they got more lipid mixing as well as more content mixing. And from there, they interpreted that the presence of cholesterol, that basically enhances the fusogenic property of GP41 fusion peptide by basically assuming a beta conformation. So they correlated the higher fusogenicity of the peptide with the beta sheet structure of the peptide. But when we started this work, we thought that influenza virus has a fusion protein called hemagglutinin, and the hemagglutinin fusion peptide is completely alpha helical. So we actually did a lot of literature search and tried to find out whether there is any relationship between the secondary structure of the peptide and their fusogenic property. And unfortunately, there is no sequence homology or sequence correlation or the structural correlation between the secondary structure and their fusogenic property. Then we asked this question, is it really due to the beta sheet structure or something else? So what we did, we are basically Fluorescence Spectroscopy Lab. So what we basically have taken two different fluorophores. So this is diphenyl hexatriene and the trimethyl ammonium derivative of diphenyl hexatriene. And this diphenyl hexatriene, it basically locates around 7.8 angstrom from the center of bilayer, whereas because of this trimethyl ammonium group, this TMA page, it is located around 11 angstrom from the center of bilayer. So we thought, okay, let's use these two probes and try to basically understand the organization and dynamics at two different length or two different depth of the membrane. So we measured the DPH lifetime, and this lifetime is a marker for polarity. So if I see reduction in lifetime, that indicates the polarity of the environment is increasing. So when we see the DPH lifetime, this DPH is located at the more hydrophobic region, and we observe drop in fluorescence lifetime. So we think that the peptide must be promoting or allowing more water to percolate in the hydrophobic region, or maybe the peptide is entering into the hydrophobic region and the peptide is more polar compared to the lipid tail, and so it shows more polarity. It basically senses polarity. So, but TMA-DPH, it does not change much because the TMA-DPH locates at the interface, and interfacial region is already very polar. And then what we did, we basically used the 10, 20, and 30 mole % cholesterol membrane, and we observed that when we have 30 mole % cholesterol in the membrane, the change in DPH lifetime is much more significant compared to 0, 10, and 20 mole % cholesterol containing membranes. So with increasing concentration of cholesterol, we could see the more drop in fluorescence lifetime of DPH, which indicates the environment or the hydrophobic environment becomes more polar when membrane contains cholesterol. But then what is the relationship of water percolation with membrane fusion? So if we look at the energy diagram of the fusion process, so what happens, these two opposed bilayers, they go through a transition state and we get a stalk. And in the stalk structure, the outer lipids, they are mixed with each other. So we can imagine a transition state structure where lipids from one membrane are basically traveling to other membrane. And when they travel through aqueous medium. So at the transition state, all these hydrophobic tails, they are exposed to water, and that's why this energy the energy of the transition state is very high. But if peptide promotes water penetration, then what happens? This opposed bilayer energy will be more than what we expect in absence of peptide. So if the energy of this opposed bilayer increases, that will eventually reduce the activation barrier and it will favor the fusion process. So we thought that whatever we are understanding from our experimental study, is it really correct? So what we did, we collaborated with Professor Subhra Ghosh in Bose Institute, and we did 100 nanosecond molecular dynamics simulation in two different membranes. One pure POPC membrane and another one was POPC and 30 mole % cholesterol. And we see in pure POPC membrane, actually the peptide, it does not penetrate into the membrane. It stays more at the head group region of the membrane. But if I have 30 mole % cholesterol in the membrane, then peptide actually enters into the hydrophobic part of the membrane. And this is the snapshot at different time points, and you can see that the peptide is not penetrating into the membrane at any time point of the simulation. But here you see from 30 nanosecond onwards, in all time point, we observe that the peptide is entering into the hydrophobic region. So what we thought that maybe because of the presence of cholesterol in the membrane, the peptide is penetrating much deeper into the membrane, and that deeper penetration of the peptide might be correlated with its function rather than the beta sheet conformation of the peptide. So, and I will come in the very late end of this talk that even the presence of cholesterol, how it can modulate without changing the depth of penetration or the secondary structure of the peptide. But we could clearly establish that it may not be solely the secondary structure, but might be the deeper penetration of the peptide is very important for higher fusogenic behavior of the peptide in cholesterol containing membrane. Then we ask that is it a common phenomena for all fusion peptides, or is it a very specific for just GP41? So what we did, we took SARS-CoV spike protein, and it was before COVID pandemic. So we worked with CoV1, and then we see that there is huge amount of sequence similarity, just only one amino acid difference in COV1 and COV2 fusion peptide. But interestingly, this COVID spike protein has more than one fusion peptide. So it has actually 3 internal fusion peptides. So what we did, we chose this N-terminal fusion peptide, we call it fusion peptide, and we chose another internal fusion peptide, and I am referring that as internal fusion peptide 1 or IFP1. And we asked this question, whether these two peptides, are they also showing same cholesterol dependence in their fusogenic property? So with that question, we studied basically lipid mixing, content mixing, and leakage. So lipid mixing provides you information of the intermixing of the lipids, and then content mixing provides you information of the mixing of 2 compartments, and leakage is a constitutive process that happens during this fusion process. So we measured leakage to basically adjust the signal of content mixing, because you lose content mixing signal if you have huge amount of leakage. And also the leakage tells you that whether your membrane retains the integrity or it gets ruptured during the process. And we use different fluorescence based technique. If anyone is specially interested, we can discuss later. But we have this tool very pretty standardized, and we measure these 3 basically observables. And when we tested this IFP1 and FP, what we observed that this is with increasing concentration of cholesterol. 0 cholesterol, 10 cholesterol, and 20 cholesterol. And same here, 0, 10, 20, blue is 0, red is 10, and green is 20. So what we observed, none of these peptides show any content mixing. So basically the fusion process is getting stuck at this hemifusion state or the stalk-like state, but it does not allow pore formation. But if I see the rate and extent of this hemifusion formation, there is a tremendous dependency on the membrane cholesterol. So if I increase cholesterol content, the lipid mixing rate as well as extent, it increases significantly. So you observe similar thing as we observed in case of GP41. And we measured all these kinetic parameter as well as the extent, and you see that for IFP it goes from 51% to 84% when we go from 0 cholesterol to 20 cholesterol, and same for the N-terminal fusion peptide. Now the question is that what cholesterol is doing here, so that we see a cholesterol dependence enhancement of lipid mixing, or rather hemifusion formation. So this is a cartoon representation that shows what I just discussed, that it shows stiff cholesterol dependence, and we published this paper in biochemistry in 2021. And then we asked that what does it do basically? And first we thought that might be the binding is different depending on the cholesterol content. And exactly we observed with increasing cholesterol concentration, the binding KD is changing. It shows very tight binding when membrane contains 20 mole % cholesterol. Whereas when it does not have cholesterol, the binding is very weak. KD value is very high. So the partitioning of peptide into the membrane is less when it does not have cholesterol. And also we measured basically homofret between tryptophan present in the N-terminal fusion peptide of SARS-CoV spike protein, and this homofret is a very good measurement for the oligomerization of the peptide or oligomerization of protein, because if 2 tryptophan comes within the Foster distance, then the tryptophan will transfer energy to another tryptophan, and any kind of energy transfer basically induces depolarization of emission. And that depolarization is manifested in terms of drop of fluorescence anisotropy. So in 0 mole % cholesterol, we did not see much change in anisotropy even in high concentration of peptide. But when we have 10 mole % cholesterol, it starts showing change in anisotropy. But if I have 20 mole % cholesterol, the anisotropy drops very drastically. So we think that this cholesterol not only allows more partitioning of peptide into the membrane, but also it promotes oligomerization of the peptide. And I told you that the oligomerization is a hallmark of any class 1 viral proteins. So the cholesterol is also promoting oligomerization of peptide, and that oligomerization is essential for its fusogenic behavior. And we measured again the DPH and TMA DPH anisotropy, and we see it does not change much of TMA DPH lifetime, but it significantly changes the TMA DPH anisotropy when membrane contains 20 mole% cholesterol. So it basically disorders the interface, and disordering the interface, other way, it will promote mixing of lipids, because now the binding force for the lipid to stay in its own membrane is less, because it is now disordered. So it will have higher probability to move to other membrane. But we don't see that kind of change when we have 0 or 10 mole% cholesterol. So not only cholesterol is promoting lipid oligomerization, but also when membrane contains cholesterol, the peptide basically disrupts the order of the interfacial region in a significant way compared to two other membranes. Then with this understanding that we have with the fusion peptides and all the literatures we had, so we thought that, okay, now we have some markers to understand that which protein or peptide, it means if it is a fusogenic, then what kind of behavior we should expect from the peptide or protein. So if we generate a peptide which shows just opposite behavior, can I use those peptides as a fusion inhibitor? So we know that all these viruses that attacked humankind, and for them to develop vaccines, it took time, and you all know that these vaccines, they basically attack the proteins or the fusion proteins of the virus. So all these fusion proteins are different, so you need a different proteins to target those fusion proteins. But even we do not know that if the strain of a particular virus changes, then we become uncertain that whether the vaccine that we developed against a certain variant, will it work for another variant. That's exactly what happens with the third phase, that we thought maybe the Delta variant, that vaccine that was good against the Delta may not work for the recent Omicron variant. So there is an uncertainty. So this one bug and one drug approach is not that adequate to respond to all these really emerging and re-emerging viral infections. So the question is, can we have a broad spectrum antiviral agent that will basically inhibit all virus and hinder their membrane entry? So to do this, either we can target the protein, the viral fusion protein, or we can target the membrane. So if we target the protein, and many strategies have been taken to target the protein, either the receptor binding domain or the fusion domain or the heptide region, there are several strategies, and there are very well written reviews and publications that talked about this inhibition. But again, they do not work when the virus changes. So what we can do, rather than targeting the protein, can we target the membrane, so that we can modulate the membrane in such a way that that protein or that membrane will no more show any fusogenic behavior. So we thought that what could be a good candidate for this. So we looked up literature and found a protein called coronin 1. It's basically a phagosomal protein, but the protein is getting recruited from the phagosomal surface when phagosome contains Mycobacterium tuberculosis. But when the bacterium is dead, then this coronin protein is no more recruited in the phagosomal surface. And this coronin 1, it basically contains tryptophan and aspartic lipids, and it basically wraps the phagosomal surface, phagosomal membrane. And there are many reasons that this phagosome evades the lysosomal degradation, but this wrapping of coronin 1 of the phagosome is one of the reasons through which this phagosome, they avoid lysosomal degradation. So we looked up at the sequence of this protein, and basically we compared the sequence along the different species, and we observed that these WD positions are pretty conserved. Then we asked that what is the significance of this WD? And those who work with membrane proteins or membrane peptides, they know that both this tryptophan and aspartic acid, they have very unique behavior with the membrane. So what we thought, okay, let's keep this WD sequence in the peptide and scramble this protein and generate different peptides. So in the first attempt, we basically synthesized 3 different peptides where the sequences are different as well as the location of WDs are also different in the sequence. And then we are try to understand that whether this peptide they can inhibit fusion or not. So we first check their binding, and these peptides they only bind to the membrane when membrane contains a negatively charged lipids. In this case, this is DOPG. So if I do not add negatively charged lipid, it does not bind. And they bind with different binding affinity. So TG has the minimum affinity, whereas the GL22 is having highest affinity. And then we check the inhibitory ability of this peptide in membrane fusion. Here, we induce membrane fusion by adding polyethylene glycol. So polyethylene glycol that basically induces osmotic pressure and it basically forcefully aggregates the vesicle and basically initiates membrane fusion. So when I say control, this control means we added 5% of polyethylene glycol to induce membrane fusion. And then we tested that after I add these different peptides and then induce fusion with polyethylene glycol, do I see any change in their fusogenic property? So what we observe basically, there is no change in the lipid mixing. So all these peptides, they basically have no effect in the stock formation. But if you look at this TG23, it basically inhibits content mixing in a decent amount. So you can look at this bar. So this is control, and then EV has no effect. GL, it blocks around 23% of content mixing, whereas the TG23, it blocks 34% content mixing. Though TG has the highest KD, that means minimum affinity. So the maybe less number of peptides are being partitioned into the membrane, but that shows the highest efficacy in inhibiting the content mixing or inhibiting the total fusion of the membrane. So we thought that what could be the reason? And we checked both DPH anisotropy and TMA DPH anisotropy, and we observed that all 3 peptides basically promotes ordering at the interfacial and hydrophobic region. There is not much difference in terms of membrane order. But when we measure the fluorescence lifetime of same probes, DPH and TMA DPH, what we see that this red and green, this EV and GL, they basically promoted water penetration. So lifetime dropped. But for TG, it increased the fluorescence lifetime. That means it blocked water penetration. So any peptide, we observe that if any peptide that promotes water penetration, that basically shows fusogenic property. And here this TG23, it is blocking water penetration. So could be this inhibition of water penetration might be responsible for the TG23 mediated inhibition of fusion. Because this works completely in opposite to what we observe for any fusion peptide. So we were very happy that, okay, this TG23, it blocks fusion as well as TG23 shows the blocking of water penetration, and we observed for fusion peptide, it promotes water penetration. So all our theoretical, our understanding of fusion with the concept of membrane organization and dynamics, they kind of matched very well. So we were very happy. But then what we did, we added now cholesterol in the membrane. So these membranes, they do not have cholesterol, it's just PCPE membrane. But then what we did, we added cholesterol to the membrane and asked that what happens to the fusion process. And you know that cholesterol is very important in several perspectives because it's a dynamic constituent of mammalian cell, and cholesterol is also a key player for entry of many viruses, and some inhibitor peptides chemically linked with cholesterol enhance membrane fusion by enhancing the membrane partitioning. So there are a lot of literature related to cholesterol and fusion, and also our literature where we observe that cholesterol plays a crucial role in fusion. So, okay, what we did, we basically prepared membrane, this PCPE-PG, and then we reduced amount of PC and we compensated that amount with PG. Sorry, we compensated that amount with cholesterol and we measured the binding constant. So binding constant is pretty much same even if I change cholesterol from 0 to 10 to 20. So the binding should not be a parameter in this case. But when we did the fusion experiment, what we observed that and for this case, we only chosen the TG23 variant. We did not take the GL22 and we discarded EV because EV does not show any inhibition. So we have chosen the best candidate, which is TG, and then we tested the inhibitory efficacy of TG in fusion of membranes containing different cholesterol content. So this data I already showed you where this TG23 reduced content mixing in a significant amount. But when membrane contains cholesterol, we see the inhibition of content mixing disappears. So this is quite interesting. So when membrane does not have cholesterol, the peptide was inhibiting the fusion. But when I have cholesterol in the membrane, then the peptide does not show any inhibition. My PhD student came and said, sir, it does not work. Then I told him that just look that why it does not work. What is the reason that the peptide is not working when membrane contains cholesterol? And this is the data that I showed you in graph that when I have no cholesterol, it's just PG membrane, it inhibited content mixing. But when I have 10 mole percent or 20 mole percent cholesterol in the membrane, there is no inhibition. So what we did, we again go back to this membrane organization and dynamics measurements, and we measured pH anisotropy as well as TMAD pH anisotropy. And this data basically shows us that when membrane does not have cholesterol, the peptide was promoting the order of the hydrophobic region. But this induction of order or the promotion of order, it reduces when membrane contains cholesterol. So this is in the membrane containing 10 mole percent cholesterol. This is in 20 mole percent cholesterol. And we see a significant drop in pH anisotropy. Similar thing we also observe in TMAD pH anisotropy, which basically probes the interfacial region. So initially TG23 was enhancing order at the interface, but in cholesterol containing membrane, the induction of order is less. And then we did our favorite experiment, which is basically the DPH lifetime that tells you the water percolation at the hydrophobic region. And what we observed that in zero cholesterol membrane, the peptide is blocking water penetration, whereas in 10 or 20 mole percent cholesterol, it does not block water penetration. Rather, it allows water penetration into the membrane. So now in 10 and 20 mole percent cholesterol membrane, these peptides, they are behaving more like fusion peptide rather than an inhibitory peptide in terms of membrane organization and dynamics result. So we check TMAD pH lifetime and there is not much change. And as I told you, this interface is pretty much polar. So you don't expect a lot of change in polarity in the interfacial region. But there is a significant difference in the hydrophobic polarity in different membrane when we add peptide to those membrane. And again, this water penetration is matching with our understanding of fusion peptide as well as inhibitory peptides. So then we asked, okay, we measured the membrane organization and dynamics, but what happens to the peptide? So whether the peptide is more dynamic in a particular membrane or it loses its dynamic property when we add cholesterol. So we measured RIS, red edge excitation shift, that basically tells you about the polarity of the environment as well as the rigidity of the environment. And this tryptophan always sits at the interfacial region of the membrane. So it is pretty much polar. So when we see change in red edge excitation shift or RIS, that basically indicates you the rigidity of that microenvironment. So when I have no cholesterol, zero cholesterol in the membrane, the tryptophan showed almost 6 nanometer RIS, whereas I have cholesterol in the membrane, the RIS is less. That means it tells the peptide is not as rigid as it was in 0% cholesterol membrane. And this data was counterintuitive of our understanding of role of cholesterol in membrane order. So we all know that cholesterol promotes order in the membrane. So if cholesterol is promoting order to the membrane, so the membrane is more ordered. So how come a peptide will be less rigid in a more ordered membrane? So what we then did, we measured the rotational correlation time of tryptophan using fluorescence anisotropy as well as fluorescence lifetime of tryptophan in 3 different membranes. And again, we see same thing. The tryptophan shows higher rotational correlation time in the membrane containing no cholesterol. But when we add cholesterol, the rotational correlation time of tryptophan reduces dramatically. So almost 0.3 nanosecond, 300 picosecond drop in rotational correlation time, which is pretty significant. So we thought, okay, so the peptide is more flexible in the membrane containing cholesterol. But then how? So we measured the depth of penetration of tryptophan in 3 different membranes. So we used a clever technique to determine the depth of tryptophan. What we did, we prepared a set of membranes and added TMA-DPH, and we know TMA-DPH locates at the interfacial region, and then we add peptide and we basically measure freight between tryptophan and DPH. So we know the freight efficiency of this partner, and then in another set of membrane we add DPH and then add peptide, and we measure freight between TRYP and DPH. And we basically take the ratio of freight efficiency between TRYP and TMA-DPH and TRYP and DPH. So if the freight ratio is more, that will tell me that the tryptophan is more closer to TMA-DPH. But if this freight ratio is less or less than 1, then I know the TRYP is more towards DPH. So just by measuring freight between TRYP and TMA-DPH and TRYP and DPH, we can determine the relative location of tryptophan in the membrane. And we observed that in 0 cholesterol membrane, the TRYP is more in the interfacial region, whereas when have 10 or 20 mole % cholesterol in the membrane, the TRYP is going little bit deeper, but still it's in the interface. Because if it is in the hydrophobic region, the ratio should be less than 1. But ratio is more than 1.5. So it's still in the interface, but in the deeper part of the interface compared to in 0 cholesterol. So we know that the head group region and the upper interfacial region are more ordered because of the hydrogen bond between the head group and the water, and also the water-water hydrogen bond. So interface is pretty much packed compared to the deeper part of the membrane. So as the tryptophan goes deeper into the membrane, it becomes more flexible. So in presence of cholesterol, what happens, the TG23 peptide, it goes little bit deeper into the membrane. And when it goes deeper into the membrane, the peptide becomes very flexible. And also it does not order the hydrophobic and the interfacial region of the membrane that much. And that's how it does not manifest the inhibitory property. Because when a peptide shows inhibitory property, the peptide has to be rigid. Because again, the flexibility is a hallmark of any fusion peptide. Because you know, during the fusion, the membrane that goes from lamellar structure to non-lamellar structure to lamellar structure. So the peptide has to accommodate itself into those non-lamellar intermediate structure. So if peptide is rigid, it will have difficulty in accommodating itself in the non-lamellar structure. So all fusion peptides, they're pretty much flexible. So adding cholesterol into the membrane, what happens? The peptide becomes more flexible, and by adding flexibility to the peptide is no more behaving as an inhibitory peptide. Rather, it behaves like any fusion peptide. So we published this result in BBA 2019. And then we measure also the CD structure, and obviously there is not much change in the CD structure of the peptide. That also nullifies that not much correlation between the secondary structure and the fusogenic or inhibitory property of peptide. You see the structures are pretty much similar. Then what we did, and this is the last part of my talk, where I will show that how it behaves in an asymmetric fusion. So when I say asymmetric fusion, that means I will study fusion of two membranes, where two membranes are having different lipid composition. And this asymmetric fusion is a much better model of fusion, because the host cell membrane as well as the viral membrane, they have different lipid composition. So we said that, okay, let's do the same experiment. Let's check the efficacy of TG23 as a fusion inhibitor in an asymmetric fusion system. So to do that, what we did, we again measured binding KD. It does not change much. So the way we measured binding KD, this is for PCPE-PG membrane. Then we prepared mixture of 1:1 of PCPE-PG as well as PCPE-PG cholesterol, where cholesterol is 10 mole%, and then PCPE-PG with PCPE-PG cholesterol, and cholesterol is 20 mole%. So we basically measured the binding KD of this peptide in the situation of our membrane fusion. So we see that there is not much change in binding KD. Then we tested the inhibitory efficacy of this TG23 peptide in asymmetric fusion between 010 cholesterol membrane and 020 cholesterol membrane. So in this case, what happened, that I added 1:1 ratio of 0 mole% cholesterol containing membrane and 10% cholesterol containing membrane, and induced fusion, and then observe that what is the effect of my peptide. And you see from the content mixing data, this is the control and this is in presence of peptide. So in 010 asymmetric fusion, peptide is still inhibitory. But if I have a 020 system or 020 asymmetric system, peptide is no more inhibitory. It's almost having no effect on the fusion. So again, we analyzed all these extent and rates, and we see that in 010 system, it's still inhibiting little bit, but in 020 system, it does not inhibit much. And even if you see in 010, the inhibition is much lower what we observed for 0-0 fusion. 0-0 means no cholesterol and no cholesterol membrane fusion. So when I add one set of membranes having 10 mole% cholesterol, the inhibitory function or inhibitory efficacy somehow diminished. Then again, we measured pH anisotropy, and we see in 010 cholesterol, it promotes order. As we know that TG23, it promotes order. But what happened here, in 020 system, it promotes order even more. So if it orders more, then we should expect that both this peptide should inhibit fusion. Because I told you that promoting order is correlated to inhibition, because all fusion peptide, they disorder membrane. But here we observe they are ordering, but they are not inhibiting. So it is totally counterintuitive of our earlier results and kind of opposes our earlier explanation. Same thing for TMADP anisotropy. This peptide promotes order in both membranes, 010 and 020. But only hope is that this DPH lifetime, what happens that when I have 010 system, DPH lifetime is still increasing. That means it is blocking water very little bit, but still it blocks water. But in 020 system, it allows water penetration. So whatever small amount of inhibitory effect that we observe, that could be due to this blocking of water. But TMADPH, not much significant change. Both of them, they promoted lifetime. They made the environment little bit less polar. But what I will focus on, that I will tell you, and which will come at the very end of my talk, that in spite of showing or in spite of promoting order, the peptide does not inhibit fusion. It did not promote, but it does not inhibit fusion. So we said, okay, let's see the flexibility of the peptide in two different membranes, and we observed in both membrane systems 010 and 020, tryptophan is showing almost similar risk, 5 nanometer and 4 nanometer. It's not that much different. There is almost similar risk. So the peptide is almost similarly rigid in both environments. And also the rotational correlation time also does not change. No remarkable change in apparent rotational correlation time. So considering the risk and the rotational correlation time, we can say that the peptide is basically almost similar. The rigidity or flexibility, whatever you say, they are almost similar in both membranes. But what happens that we observe certain difference in their fusogenic behavior. What is the difference? Now in 010 system, we observed certain amount of inhibition. Less, but we still observe certain amount of inhibition. But when we observe the same thing in 020 system, there was no inhibition at all. But if I see the membrane organization and dynamics parameter, other than DPH lifetime, all parameters for 020 system supported its inhibitory character. Even it supported the inhibitory character, but actually it did not inhibit. So we thought, okay, depth of penetration is also same. There is not much change in the depth of penetration. So then we asked that what could be the difference? What is the difference, or why this TG23 is behaving differentially in 010 and 020 mole% membranes? One is 010 and another was in 020 system. Though the membrane dynamics and order parameter supports, they should behave similarly, other than DPH lifetime. They should behave similarly. So then we see that if you look at the cholesterol structure, it has a small OH head group with a bulky hydrophobic tail. And that tail is very different from the phospholipid tail. But it has overall an intrinsic negative curvature. When I say intrinsic negative curvature, means it has a small head and bulkier tail. So it has this kind of inverted cone-like structure. So anything that has this inverted cone-like structure, literature says that those lipids, they have the fusogenic property. Because when you see that stalk intermediate basically forms due to the intrinsic negative curvature formation in two membranes. So if I have lipids that has intrinsic negative curvature, that will help to induce negative curvature in the total membrane, and that way it will promote fusion. So we thought it could be due to the negative intrinsic curvature of cholesterol itself that has fusogenic property, and could be that fusogenic property is basically superseding the inhibitory efficacy of TG23. Because TG23 in 020 system supports the all membrane organization dynamic study supports it should inhibit, but it does not inhibit. So then it could be that the effect of cholesterol is superseding the effect of the peptide. So what I tell you, this is a quick summary, that in 00 system it inhibits, 1010 system it inhibits partially, 2020 system it does not inhibit, 010 system it inhibits partially, 020 system it does not inhibit at all. So this peptide induced water penetration, it is directly correlated with its fusogenicity as well as inhibitory role. But at the same time, the presence of cholesterol and the direct effect of cholesterol in the membrane fusion, apart from its role in altering the lipid-peptide interaction, the direct effect is also a very important parameter to study membrane fusion. So this membrane composition is an added complexity in the journey of developing peptide-based membrane fusion inhibitors. So when I said beyond, that beyond part I need to focus, that don't just look in the sense of lipid-peptide interaction, but also those peptide or also those lipids, they have direct involvement in the process. And that direct involvement we sometimes miss when we look or when we focus our work mainly on the lipid protein or lipid peptide interaction. But we should be also careful about the direct impact of lipid structure and shape on the fusion process. So the take-home message is lipid composition modulates the membrane peptide interaction and tunes the fusogenic or inhibitory ability of peptide. But lipids have their own story to tell. The shape of lipid has direct impact on membrane fusion. And finally, I would end with acknowledging my students. So I mainly discussed about the work of Gitanjali and Gaurav, who recently left the lab after graduating from the lab, and my collaborators. So this work I majorly collaborated with Subhra for the molecular dynamics simulation, and Professor Surajit Bhattacharya in NTU with some NMR structure. And we are now doing a study with Joydeep Basu in IISc, looking at how lipid dynamics changes on the membrane. So we know about these domains structures, but we are now seeing that how presence of peptides changes the movement of lipids in a particular domain. And I should thank all the funding agencies like ACRB, STAR, Odisha DST, and UGC. And thank you so much. Sorry, I took a little bit more time, I think.
No, it's fine. Thank you, Hirak, for the clear, lucid talk. So we can take questions. Anybody? Professor Mishra.
Okay, we are proud of Hirak. He has so vividly explained the work. So there is nothing is unanswerable or nothing is unquestionable. But again, for a basic question, a layman question, cholesterol, it plays a vital role in lipid structure, both for this flexibility, rigidity, and structural change, shape change. And also we explained how this cholesterol is involved in this fusion. Have you any idea that this how this cholesterol interacts with protein directly, molecule to molecule, like let us say in molecular docking or something, so that it can change the protein structure?
So in the molecular dynamics simulation, we tried to find out the basically time spent by lipids near the peptide. So we tried to see whether the cholesterol is spending more time near the peptide or not. But we did not find any significant difference between other phospholipids and cholesterol, which is basically retaining more time near the peptide. So or the experiment, but definitely I cannot conclude with just one MD simulation. I need to try with other peptides.
Like molecular docking, if cholesterol, how it is entering into the, or let us say how it is interacting with the protein directly?
Actually, docking will not give you much information because when we dock, we lose, or actually we ignore many interactions.
Why I have asked, you can take, let us say, cholesterol is changing the structure or polarity and flexibility of your lipid membrane. You can take another model without cholesterol, which can do the same thing.
So that's exactly what we did with the molecular dynamics simulation with pure POPC membrane and POPC cholesterol membrane. Yeah. And in the POPC cholesterol membrane, we basically measured that how much time a POPC lipid is staying near the protein or near the peptide, and how much time a cholesterol is spending near the peptide. So there is a beautiful simulation work by Durba, and he showed with 5-HT1A receptor that this retention time or time of any lipid, in that case they showed cholesterol, the cholesterol is staying much longer time near the protein during the simulation. So that way it says that there might be some specific interaction, but we did not see that kind of thing.
Okay. Thank you, Hirak, for a nice talk.
Thank you. Shumon?
Again, very interesting work, very detailed, and you have done a very systematic analysis, very interesting, and lots of complexity. I had a more basic and naive question. How does the cholesterol affect the surface tension of the membrane? Has there been any measurement of the surface tension?
Yes. So it basically increases the surface tension. So surface tension increases. Also, there are AFM study that tells you that how much force you require to bend. But when you measure the AFM, basically, no. So I told. So just let me tell you first the AFM experiment. They showed that if you want to bend the structure, and if the membrane contains certain amount of cholesterol, then bending is much easier.
Okay.
It's much easier. And also there are literature with they actually did this neutron scattering experiment, and they showed that if you have cholesterol in the membrane, that promotes hexagonal phase much easier than if you do not have any cholesterol.
Okay.
So it basically promotes hexagonal phase, as well as you require less energy to bend the membrane. But surface tension, I don't know. I don't know literature regarding the surface tension.
So if, I think then maybe the surface tension should decrease, because
it should decrease.
If it is more flexible, then probably it is becoming lower, because otherwise it will become more rigid. If the surface tension is higher, it will become stronger.
It should decrease. It should decrease.
Yeah. And another thing that I think you, so basically I'm trying to understand, so one is the mechanical strength of the membrane. So when you are talking in terms of the surface tension and curvature, but also there is thermodynamically, I think the polarity is also increasing, what you have found based on your lifetime. So there is this, basically I'm trying to understand that there will be both a kinetic aspect, that when something is entering, that there will be some kind of a barrier first, like you showed a free energy profile. So there is a barrier before, let's say, the stock formation, and then there is a minimum. So there will be a stability difference, that is the thermodynamics, but also there could be change in the barrier, which is the kinetics. So did you kind of look into this separately, the difference? Is there any kind of difference in the trend of the kinetics versus the thermodynamics?
So recently we started, actually it's long back during my postdoc, what I did, I basically developed a kinetic model, analytical model based on these three observables. So there's lipid mixing, content mixing, and leakage. So you fit this observable in that model and you get the rate constant of each intermediate formation. So recently we started analyzing all this data in our model to really get an idea about this rate constants of each state. So that basically tells you the kinetic, that only gives you the kinetic information. But when we do any fluorescence measurement, all these fluorescence measurements are in the steady state. It's in equilibrium state. So whatever water penetration we observe, that's basically the thermodynamic. So I do not know what is happening in the intermediate structure, but at least in the closed or the apposed bilayer, in the apposed bilayer, you are starting with little bit more water into the hydrophobic region. So the energy, overall energy will be high, and if that energy goes up, eventually you reduces the activation barrier. That's the hypothesis.
Interesting.
And this is the only parameter I find in the literature, because I have been trying to find out one yardstick that will tell you that this peptide is fusogenic and this will not be fusogenic. So that this is the only parameter that water penetration in the hydrophobic region, it is kind of consistent. But other parameters, they change from peptide to peptide and even lipid composition to lipid composition.
And just a quick technical question: how do you measure the lipid mixing? What is the?
So what we do, we prepare a vesicles, and in that vesicles we keep a fluorescent donor as well as a fluorescence acceptor. So we use NBD and rhodamine. So NBD is tagged with a phosphatidylethylamine and rhodamine tagged with a phosphatidylethylamine. We put them in the same vesicle. So now both the donor and acceptor, they are close to each other. So you see highest freight. Now you mix this membrane with unprobed membranes.
I see.
Those membranes do not contain any donor or acceptor. So if lipid mixing takes place, what you will see, your freight will reduce. So we just follow the donor fluorescence. So at the beginning, donor fluorescence is minimum, because donor energy has been transferred to the acceptor. And with time, if mixing takes place, this donor and acceptor, they will be separated because other lipids will enter and this probe will go to other membrane. So the distance between donor and acceptor will increase. So that will increase basically the fluorescence of the donor.
Very interesting. Yeah. Okay. Thank you. Yeah.
Thank you.
Any other question? So maybe Hirak, I asked you something that regarding the mechanical strain of this membrane. So whenever the membranes are fusing, there is a change in the curvature. So what is the driving force of that the curvature should change and they fuse?
So in biology, the totally curvature change is induced by the protein. Because when this fusion peptide goes into the target cell or the host cell, then you have this 6 helix bundle formation. So this 6 helix bundle formation that basically pulls two membrane close to each other. So when you pull from one point, it will automatically form curvature.
Okay.
Isn't it?
So why they will come together? I mean, come close or approach together.
So basically, you have multiple proteins definitely, and you are bringing them together, but still your contact point will not be as large as you imagine because the viral virion surface is much smaller than the host cell. So it's always like a flat membrane and the virion is sitting on it. So your contact region is very small.
Okay. So it's a kind of driven process rather than a random process.
It's a driven process. And what is shown is that lipids like phosphatidylethaноlamine, cholesterol, they basically promotes this process because their intrinsic negative curvature helps the membrane to attain that state, that curved state. So in model membrane, say for example, where we are not looking at flat surface and curved surface fusion, both are curved fusion. But if I add phosphatidylethaноlamine, in recent 2021 itself, we published a paper in JPCB, and we showed if we have 30 mole % phosphatidylethaноlamine in a membrane, then it forms the contact or it forms that stalk structure very easily. So if you see the overall structure, it's a spherical. But the presence of this negatively charged lipid, it creates this local inverted or local negative curvature or local curvature into the membrane.
Okay, so in any lipid or without any lipid, suppose there are some vesicles, so they do not usually collide and mix or exchange things, no? It is always driven.
They are stable.
Okay.
So they are stable. Okay. So they will be stable for 3 to 4 days.
Okay. But ripening is, I mean, or Oswald ripening kind of thing, they do occur or they don't?
It's very rare, because you have those structured water in between, and they don't allow the membrane to coalesce with each other.
And does it also depend on the charge of the membrane that they have? If they have charge, then they will repel each other.
So when you have charge, most of the time what happens, the charge is being neutralized by the charge of the fusion protein. So if the membrane surface has negative charge, then definitely we will find positive charge in your fusion peptide or fusion protein. Otherwise, it will repel each other.
Exactly. Okay, thank you. So any other question from anyone? Okay. If not, then let's thank Hirak for a beautiful presentation. And thank you, Hirak. I mean, we are very unlucky that we cannot welcome you personally, but sometime you must come.
Yeah. Thank you so much. Thank you. Sumon. Thank you.
Yeah.
Thank you. Yeah.
Thank you.
Yeah. Take care. Stay safe.
Bye.
Yeah.
Bye.
Bye.